Hey guys, it's Mr. Citron here, and we're going to do a little article review, and we're going to do the article from The Scientist, which is a publication that is really designed for not the general public, but the educated public. So it's a little bit higher than your average like USA Today or Canton Repository. It does talk about research that is going on. But it is not a research journal. It is a summarization of. And this one was from February 16th, 2021. So just a few days ago, really. Today is the 28th. So we're talking just 12 days ago that this was published. So, and going into the topic that we're going through, I really feel like this particular one, talking about the coronavirus, which is the official name is Sars Cov2. And the fact that we know a little bit about mRNA, we know a little bit about proteins, we know a little bit about DNA, this makes a little bit of sense. So I'm going to read it for you so that any of the larger vocabulary is there. And if you want to, you can follow along. If you prefer, it makes it a little bit easier. So from February 16th, so SARS-CoV-2 with genomic deletions, escapes, and antibody. Researchers identify deletions in the N-terminal domain of the spike protein that allow the coronavirus to avoid antibody neutralization that may contribute to the emergence of new variants. What that means in that blurb article, that subtitle, is that with a small mutation in the protein, which is what we're doing this week, the coronavirus no longer will be neutralized or will not be destroyed by antibodies that your body creates. And that factor can contribute to this new variants coming on board that we are now experiencing in the world. So that's just a quick introduction there. When SARS-CoV-2, the virus behind COVID-19 pandemic, first emerged, scientists expected it to evolve slowly because the virus copies its big RNA genome with a polymerase, that's an enzyme, that also corrects errors, thus minimizing the chance for certain types of mutations. This enzyme functionality isn't present in other RNA viruses such as influenza and HIV, which causes AIDS, which accumulate single nucleotide polymorphisms, where one nucleotide is substituted for another much more quickly than SARS-CoV-2 does. So in other words, a polymorphism is when one single base pair, an adenine, a thymine, a cytosine, a guanine, or in this case, because it's RNA, adenine, uracil, cytosine, and guanine, one gets popped up and out and gets replaced by a different one. So where there once was a U, now it is an A. Where there once was a G, there is now an A. Where there once was an A, there is now a, whatever the case may be on one side of it. You know that if you change one codon, one base pair of that codon, you can then change the amino acid that gets coded for. So, rereading this, the enzyme functionality isn't present in other RNA viruses such as influenza and HIV, which accumulate single nucleotide polymorphisms where one nucleotide is substituted for another much more quickly than SARS-CoV-2 does. In other words, we didn't expect COVID, SARS-CoV-2, to actually mutate. This genomic stability was thought to be good news for vaccine design because it became apparent in recent months with the emergence of these two variants, the B.1.7 and the B.1.351, and the other variants, that SARS-CoV-2 is now mutable, meaning it can be mutated, and might put up a fight against neutralizing antibodies that we create in our bodies that attack or go after the spike protein that is on the outside of the virus. In a study posted as a preprint in November and published in February 3 in Science, researchers showed that SARS-CoV-2 tend to delete portions of the RNA. Whole portions get deleted out that encode the N-terminal domain, NTD, which is going to be referenced later on to the article, of the spike. The N-terminal domain of the spike. Don't worry about what that means, but essentially what it means is that a portion of that protein spike gets deleted, so that means the amino acid sequence is different, and therefore we have a mutation. And this is a mutation that its proofreader can't detect. And these findings and those of other groups offer an explanation for how new SARS-CoV-2 variants arise and escape antibody recognition by the host immune system. You are the host in this instance. And as we all know, hosts, parasites, or infection material, they don't want to kill the host. The host, that's not the idea, to kill the host. The idea is to live off and to survive the host. So evolutionarily speaking, keeping a host sick is better than killing a host. Now, a little bit on to the deletions here. So deletion, which is this type of mutation, represents a way to rapidly alter an entire stretch of amino acids and more rapidly alter the structures those amino acids are contributing to, says co-author Kevin McCarthy, a virologist at the University of Pittsburgh. These mutations could accelerate SARS-CoV's evolution and then complement mutations that it's already making. So when you do a full deletion, it changes that whole amino acid sequence by popping it out. That could be deleterious, that could be advantageous, but either way, it's going to be a mutation. It could be benign too. We don't have no idea if it's going to happen or not. So, all right. So the project started when two physicians at the University of Pittsburgh's Medical Center contacted Paul Duprex, a virologist, and the director of the Center for Vaccine Research at Pitt. The doctors had an immunocompromised cancer patient, so their immune system was not functioning properly, it was compromised, who passed away after a SARS-CoV-2 infection that lasted for 74 days. And the infection did not respond to the common treatments, including remdesivir and dexamethasone. So Duprex and his team took a look at the viral genome to see if they could figure out why the infection had lasted so long. From samples aspirated, taken out of from the patient's trachea 72 days after the initial COVID-19 diagnosis, the researchers isolated viral RNA and several SARS-CoV-2 variants, which likely evolved from one initial infection during the illness, and sequenced the S gene, which encodes the spike protein. So this whole gene, this long segment of DNA that codes for information, they decoded the whole thing. They found two variants that contained different 3 nucleotide deletions in the NTD of the spike protein. And just as recall, NTD is the the N-terminal domain, NTD. So they found two variants that contained different 3 nucleotide deletions, just 3. So an A, a T, a C, just 3 of them. Sorry, I said T again. An A, a U, a C. Deleted that. There's no T in this. It's RNA. And they deleted those 3. There were 2 differences that contained 3 nucleotide deletions. That's it. So all we did was adjust that amino acid sequence by 1. Duprex mentioned the findings to McCarthy, who, with the help from collaborators, combed through the nearly 150,000 SARS-CoV-2 sequences that had been deposited by the end of October 2020 in the Global Initiative on Sharing All Influenza Data, GISAID database. The researchers found 1,108 viral genomes that had deletions of varying lengths in the S gene, 90% of which were located at one of 4 sites in the NTD. There are how many different genomes that had deletions of varying lengths? So we have found 1,108. Oops, hit the wrong button. Actually went to just say it. Let's go back here. So we have found that many. So that means that there is conceivably over 1,000 different variants to SARS-CoV. Many of the deletions were in patient samples that appeared to be persistent infections, similar to the one the Pittsburgh patient experienced. And some of the most concerning new variants, including B.1.1.7, initially identified in the UK, and B.1.351, first spotted in South Africa, commonly referred to as the British variant and the South African variant, have 1 or 2 of these short deletions in the NTD, along with a cluster of mutations in the receptor binding domain. Receptor binding domain is the RBD of the spike. The receptor binding domain has to be there for the virus to attach to your host cells in your respiratory system. So the cell membrane of your host cells has to be able to understand the receptor binding domain, which is essentially a form of protein. Those are those transport proteins, those surface marker proteins that we referenced when we did the cell membrane. All right, here we get a little picture of the spike protein. what we're looking at here. And here's a little, so deletions, this is the explanation of the graphic. Deletions in the Sars Cov spike protein sequence, it's the horizontal blue bar down here, affect the shape of different parts of the protein. So let me zoom out my screen so we can see this a little bit together. So wherever we see these proteins, whatever this bluish, they call it cyan, the bar graph on the left shows the relative frequency of the recurrence of, gotta scroll up, sorry, deletions in the correspondingly colored parts of the spike protein, and deletions tend to occur out, looks like I cut off, to occur outside. So this is where we're getting the mutations, and then these are the variants and those colors that occur on the edges of that protein. So it's kind of a zoom in of this sequence comes over here. Okay? Let me make that bigger so we can read it more carefully. The researchers then tested whether 6 of these deletions would disrupt antibody binding. In other words, can your immune system bond to it? 3 of those deletions and 1 deletion combination in the NTD eliminated the ability of an antibody previously isolated from convalescent patients to neutralize the virus. So it is no longer able, it eliminated the ability of this antibody that was harvested from patients, convalescent patients. Convalescence means that they're in recovery, to neutralize the virus. So 3 of those deletions and the 1 deletion combination in the NTD made it so that your body cannot fight off. It is no longer immune. So this specific antibody completely lost its effectiveness, but convalescent serum, which contains a full set of antibodies, not just one, did neutralize each of these variants. So that's a good thing. So it fights off one specific antibody, but when you have an immune response, you don't just make one, you make a whole series of antibodies. And although this mutation made one antibody unable to treat your disease, the rest, if you have your full complement, you're okay. Antibodies are, quote unquote, quote, swarms looking at different shapes of different parts of the molecule. So it's not an all or nothing whenever we're thinking about the swarms of antibodies. But it absolutely is all or nothing when you're thinking about this particular antibody, which binds at this particular space, explains Dubrex. So back to our NTD deletion variants and the vaccine potency. So how potent is our vaccine going to be if we have this NTD deletion? The next steps are to catalog the effects of these deletions on large collections of plasma samples, including from survivors and vaccine recipients. Theodora Uhatsa Ano, I don't know if I pronounced that right, a virologist at the Rockefeller University in New York, who is not part of the work, tells the scientists. So we often, the scientists, they often quote people who do not do the work as a peer review. So they are doing the analysis much like you and I are doing that analysis, only they are far more educated than you and I combined. So she says that this could help answer questions about vaccine efficacy, reinfection, and if vaccine design should account for these deletions. These mutations are, quote, just a part of the variation seen in the spike at this moment. So what do we got here? The Moderna and Pfizer biotech vaccines, those are the 2 most common ones, which are the only 2 approved for use in the United States. As of Saturday, the Johnson & Johnson, J&J, got approved. It's a single dose shot. So this article, although is very current, does not have this weekend's updated information. Both include RNA encoding the full-length spike protein. So that's what so once injected into the person, the RNA is translated into promoters, and the recipient's immune system then responds to and develops a memory of the encounter. So this is how a vaccine works right here. And this is what we're doing, and this is what I'm getting on Monday. This is what I got a month ago. This is what everyone who's in Ohio and anyone in the United States has gotten. We've injected a section of the RNA to create a protein. So the RNA goes to the ribosome creates a protein. We're skipping all of the other steps and jumping right to protein production, which then creates an immune response that our body then catalogs. And according to the COVID-19 vaccine candidate tracker published in February 12th by the World Health Organization, 11 vaccines that initiate immunity to just the RBD of the spike are at varying stages of clinical trials. Quote, the message to the vaccine world is that it was the correct choice to use full-length spike, and those others where that got cut off, based strategies, only the RBD are missing. Parts of the protein, in this case, the rest of the spike, particularly NTD, which patients have been shown to mount an antibody response to, says Gregory Ippolito, an immunologist at the University of Texas at Austin, who was not involved in the work. Ippolito and his colleagues, part of a team led by George Giorgio at the UT Austin, posted a preprint in BioReview in December showing that more than 80% of the antibodies to portions of the spike in 4 convalescent patients bound to domains other than RBD. The vaccines that leave out portions of the spike don't give recipients' immune systems the chance to develop full complement of antibodies. And this is really the key here. I'm going to pause it right here, or at least explain this here. So if you get a vaccine that leaves out portions of that spike protein, but that portion of the spike protein can mutate with a deletion, you lose the full complement of antibody production. And therefore you lose or risk becoming a less effective vaccine. So it was a good thing that the original development, so it was the correct choice to use the full length spike. Instead of just a little tip of it or just a little section of it, they gave the mRNA that's being injected into my shoulder is a full complement of the whole genome, which I think is somewhere around 29,000 base pairs. But we're going to look at that this week as well, because I'm going to get you the whole sequence. I found the database. I'm going to get you the whole sequence for COVID-19. The study also points to considerations for the therapeutics that give passive immunity, such as antibody cocktails, said Jason Lavender, another member of the team at UT Austin. It's important to include in those cocktails not just the RDB-directed antibodies, but also the NTD-directed antibodies. So essentially, you're ingesting those or injecting those. Cocktail is often a way we call it when you're putting together an oral pill of medicine. So including all of those things so that your body can incorporate it straight into the antibody step. It might be injectable in this, they don't say specifically, although I would think that if it's an antibody, it has to be injected. Right, so some keywords in here and some of the citations. So much of this should be accessible to you. I know that this took me 17 minutes to read it. It's only a 2-page article, but well, kind of a 3-page article, but with a big picture in the middle. So hopefully that is helpful, and as you read through it, you can then understand it. And I'm going to post some discussion questions for us to work our way through for this particular assignment. Thank you for your attention. Have a great day.